Alzheimer's disease is a fast-growing health problem in the United States and across the globe. As populations continue to age and lifespans increase, more individuals are living long enough to develop this progressive neurodegenerative disorder. As a result, more families, caregivers, and healthcare providers are feeling its impact, emotionally, socially, and economically. Because Alzheimer's has such a big impact, it's a key area where scientists are trying to develop new and upcoming treatments. One growing area of research, which we'll discuss more in depth today, involves something incredible: the human genome. But before we start on that conversation, it's important that we cover the fundamentals. Our brains are made up of billions of neurons, all coming together to form the control center of our lives. Researchers have pinpointed two major brain changes related to Alzheimer's. First, the accumulation of a protein fragment called beta amyloid outside of neurons. And second, the accumulation of an abnormal protein type called tau tangles inside of neurons. Both of these elements cause inflammation, impaired metabolic function in the brain, and overall dysfunction. Research has shown that these proteins, or biomarkers, may be detected up to 22 years before the onset of Alzheimer's symptoms. Once these symptoms begin, some examples include difficulty remembering events or information, emotional apathy or general depression, difficulty with communication or speech, and in later stages of the disease, slowed walking, speaking, and swallowing. The FDA has approved some pharmacological treatments for slowing the progression of Alzheimer's disease. These have been proven most effective in mild cases, and little data has been published on the potential in moderate or severe cases. It's also important to note that current Alzheimer's treatments can help.
Before looking at how CRISPR can be used to treat different neurological diseases, we wanted to start by breaking down what CRISPR is and how it works. CRISPR actually refers to a part of the DNA which contains a bunch of repeats, meaning it has the same letters or sequence over and over, and a highly variable region, meaning the sections are very different from each other. With the CRISPR sequence, so this part of the DNA, there's often something called Cas proteins, or CRISPR associated. That just means that these proteins are right next to the DNA in the sequence. Together, they form a complex that acts a lot like molecular scissors. It's able to go and cut the DNA where we tell it to. What's important to note is that this whole system is guided by something called the guide RNA. How this works is the guide RNA acts a little bit like the line leader. So this guide RNA attaches to the CRISPR complex. Remember, that has the Cas proteins and the CRISPR DNA. And then this guide RNA is able to take the complex to wherever you code it to, and it's able to force the CRISPR complex to cut the DNA in that location. So let's say there's a section of the human DNA that you want to cut. All you have to do is add a matching guide RNA to that same sequence into this CRISPR complex, and then the guide RNA will take CRISPR to that exact part of the human DNA and cut it there. Now, it's important to note that this cut doesn't always work perfectly. Sometimes there are little mutations, or we say indels, meaning that there are different bases that were inserted or deleted that weren't really supposed to be there in the first place. This is really important when we're thinking about implications in terms of gene editing in humans. So yeah, CRISPR, pretty straightforward. It's just a giant pair of molecular scissors that is guided by a guide RNA to certain parts of the genome into the DNA, where it's able to make cuts wherever we want.
Now that we've discussed what causes Alzheimer's, it's time to talk about how CRISPR is being used to identify new treatments. Recent research has looked at a couple of different genes to target, and today we will focus on two of them, PSEN1 and APOE. PSEN1 supports an enzyme called gamma secretase. This enzyme breaks down proteins, and this degradation leads to beta amyloid plaque formation. If we can fix these mutations and decrease enzyme activity, then this will prevent the breakdown of AB proteins, decrease AB peptide formation, and decrease plaque formation. That is exactly what Constantinides and his group did. They focused on a mutation that led to increased PSEN1 function, which leads to early onset Alzheimer's. Using CRISPR-Cas9 systems, they were able to fix this mutation, decreasing function of the PSEN1 gene. This group was even able to perform this experiment on real human fibroblast cells, and they saw a decrease in total plaque formation. The APOE gene regulates the calcium concentration in neurons, and it can take on 3 different forms, including E2, E3, and E4. E3 is the most common form and results in normal calcium regulation. E2 also results in normal calcium regulation. Expression of E4 leads to an influx of calcium ions in the brain, leading to increased cell death and also increased tau phosphorylation. Wadhwani et al. Were able to use targeted CRISPR systems to edit the E4 version into either the E2 or E3 version of the APOE gene. In human cells, their edits let cells withstand induced cell death and decreased overall tau phosphorylation. These are just 2 examples of how CRISPR is being used to slow down Alzheimer's progression. There are many more techniques being worked on even today. And in the future, scientists hope that using cutting-edge technology like CRISPR will slow progression further and potentially cure the disease entirely. One day, CRISPR very well may make Alzheimer's a preventable ailment, saving the lives of hundreds of thousands of people.
When discussing any technology, especially in regard to medicine, it is essential to understand the societal and ethical implications of its use. With CRISPR, these implications can be divided into 4 main realms of concern: individual autonomy, eugenics, future consequences, and treatment versus enhancement bioethics. Firstly, individual autonomy and informed consent. With genomic editing being such a rapidly advancing technology, it is essential to understand the consequence of being subject to it. A major concern for bioethicists is that people are not adequately informed on such and are therefore unintentionally subjecting themselves to something they do not want. Secondly, in regard to eugenics, the fear that such technology is only available to the wealthy developed world, especially when those who would benefit most from it are the opposite, remains prominent. With germline editing, the idea of entire lineages being rid of certain diseases and at a higher chance of preventing others creates a societal divide borderline dystopian. Disability advocates explain that such a practice of classifying certain genes as good and certain genes as bad determines an individual's life as less worth living on the basis of their DNA, therefore ostracizing communities that already face challenges in society. As for future consequences, since it is a new and developing technology, the off-target effects and unwanted on-target effects can be anticipated through systematic analysis, but are not known. Lastly, while it does not pose an ethical dilemma for the medical use of CRISPR, the use of genome editing for bodily enhancement in an individual or their offspring is a highly debated topic. Most agree that the aesthetic use of such a technology is unnecessarily risky and opposes nature. While it is essential to recognize the reality of all of these implications, in the context of a potential Alzheimer's treatment or cure, they are surpassed in all societal realms via a risk versus reward analysis. Scientific researchers with extensive education on the mechanisms of this science, theologists who specialize in a wide array of religious thought, philosophers who emphasize the need for dignity and balance, and the general public ignorant to the workings of genomic editing have been found to agree that the use of CRISPR can be justified in therapeutics if the resulting effects reward outweigh the potential risks. With the state of the current technology and off-target effects being a real possibility, and for the future consequences being relatively unknown, such risks are high. For diseases that have alternative treatments, cures, and preventative measures to CRISPR-based editing, the analysis of risk versus reward is very complex. In the context of Alzheimer's, which has no cure and extremely limited symptomatic treatment, it is much simpler. To explain it, without CRISPR interference, the individual would continue suffering from the disease, and their quality of life would worsen as it hinders their ability to function. With successful use of CRISPR technology to correct the genetic causes of Alzheimer's, which are not yet known, the individual could have a chance at full or partial recovery. This chance is enough to convince the major societal divisions, theologists, researchers, philosophers, and the general public to favor CRISPR-Cas9 system-based Alzheimer's treatment approaches. It is essential to reiterate at this time the need for informed consent for all bodies, which is one of the only areas in this bioethical analysis that limits the favorability of this approach. With individuals diagnosed with Alzheimer's and other dementia-related diseases, the ability for them to comprehend the vast risks and choose to subject themselves to such can be difficult. As long as it is accounted for appropriately, then there are no strong ethical oppositions to the use of CRISPR or genetic editing techniques to treat and cure Alzheimer's.